be distracted. In fact, they found that people learn better when they're being assaulted by a variety of information, because the brain deals with that assault by trying to pull out the relevant information and learn it. So, I mean, best place to study, I guess, is a bar. You did not hear that from me. Okay. Yeah, you're going, yes, I did. All right. All right. So what I was trying to get at here is that, so short term, we're dealing with transient modifications at pre-existing synapses. In other words, that's the rate of firing. Whereas with long term, we're looking at structural changes. All right. Actual changes within the relationship between the cells. Okay, moving away from memory, I want to talk about language. All right, we actually have two areas in our brain that are responsible for what we would think of language skills. All right, now, for most of us, our language centers are located on the left side of our head. All right, there are some of you in this room, about 1 to 5%, I can't remember what the number is exactly, that it's reversed. Who it is of you, we don't know. Insignificant. All right. But what we have is we have two areas. We have the area that's called Broca's area and the area that's called Wernicke's. Here's Wernicke's back here in the back. The temporal lobe, Broca's area is up here in the frontal lobe. And they're real easy to remember, especially if you're bilingual and you speak Spanish. All right. If you speak Spanish, who can tell me what the name for mouth is? Boca. See, everyone here? Boca. All right. Notice one of these sounds or looks like boca. It's Broca. And Broca's area governs speech. Speech, if you'll notice, is the movement of lips, tongue, and mouth in order to make certain noises. It is a motor skill, which is why it's located up here in the frontal lobe. So speech is Broca's area. All right? Articulation, or mumbling, or whatever it is that you want to do. Wernicke's area, on the other hand, is not about moving the lips. Wernicke's area is about understanding language. Most of you in this room can understand the words that are coming out of my mouth. All right? That information goes, you hear the sound coming into your ears, you take these sounds and you convert them into something that you understand. When you read a book, you look at words on the page and you translate those words on the page into something that you understand. And then you use that as language. So it's language comprehension. It allows you to understand language. So Wernicke's deals with understanding language. Broca's, I should do it on this side since it's on the left side. Broca's is about articulation of speech. Now, if you have a lesion, for example, in Broca's area, all right, so we have a lesion up there. That person with that lesion would be perfectly able to understand you if you ask them, are you having a good day? Their response would be something on the lines of, all right? Because they can't control their mouth to articulate what they're trying to say, but they can understand you. A person with a lesion in Wernicke's area would say, are you having a nice day? And they might say, apple, banana, train. I mean, it's funny, but literally, when you start doing your rounds and you start going through where you have patients who have had strokes in these areas, it's the most odd thing in the world, where they look at you as if you've said a perfectly reasonable thing and they come out with a perfectly unreasonable response. Because this allows you to understand what is being asked of you. And just to put it to a test, let's see if we can understand language. What do we have here? I can see you. It's not I, battery, water, cow. Yeah, and you can tell this is a U because you can see its udder. See? Yeah. Sheeps have udders. You know. But, you know, many of you were sitting there going, okay, I'm not sure what these hieroglyphs are, but many of you who play puzzles and stuff like that were able to recognize this because you did a conversion from image to homonym. And then you put the homonyms together and said, oh, wait a second, that sounds like something that I know. That's purely Wernicke's area. And then you, as you're sitting there saying it out loud, that's your Broca's area allowing you to articulate the words as you're trying to, I I can see you. Okay. So that's the exercise to try to demonstrate these two areas. So we're leaving the cerebrum, and what we're going to be doing now is we're going to be talking about, since we understand that the central nervous system is responsible for integrating information and processing it, what we want to look at now is what type of information are we giving to the central nervous system. And so they're talking about the afferent information. And whenever you see afferent, you can just think information that's coming into the body or coming into the central nervous system. And there's actually two different types. We have visceral afferent information and we have sensory afferent information. For the most part, we more or less ignore these. You know, when we think of afferent information, we think of, and you even heard me say, the stuff in the outside world, what's coming inside. But visceral information is the input that's derived from our viscera. All right? This is the stuff that our body uses to understand whether or not we are maintaining homeostasis within the body. And it's done at a subconscious level. For example, right now, you do not know whether or not your water-salt balance is in balance. You can't say to yourself, aha, I'm 296 milliosmoles of solute right now in my blood. I need to drink. Well, instead, what you do is any sort of conscious awareness of your visceral input is a result of the processing, right? It's the integration. And so you'd be saying, for example, I am thirsty, or I have too much and so I need to go to the bathroom. Too much fluid. So visceral input, or I'm hungry, is subconscious. You're not aware of what your body's actually monitoring, other than the fact that you understand your body is monitoring it, right? Anyone here aware of what your blood pressure is right now? Of course not. I mean, some of you are a little bit more like this, and so your blood pressure is dropping. That might be conscious awareness of that you're sleepy, but you don't know what your actual blood pressure is. The other type of afferent information, sensory afferent, and this is the sensory input that's coming through, and this is the stuff you're actually consciously aware of. So we have two different levels, or two different types of this. This is the somatosthetic. Remember, that's the touch, the pressure, the heat, anything external to the body that we can become aware of. And then we have what are also referred to as the special senses, which we're going to spend time talking today about smell and taste, but includes also vision and hearing, which we'll spend time talking about tomorrow. Yeah. Somatosthetic, touch. Something is sticky. Something is hot and, wet. That's somatosthetic. It's being aware of the pressures and touch and stuff that you're feeling from the external environment. Now, in order to get these sensations, you need something to be able to become aware of that stimuli. And so what we have are receptors. And receptors are located on the peripheral end of afferent neurons, and there's many different types of these afferent neurons, and these are just some examples of them. And the job of the receptor, simply put, is to convert the energy of the stimulus into the energy that the brain understands, which is an action potential. All right. So what we're trying to do is we're trying to convert all these different types of stimuli into an electrical signal. All right. Because it's the electrical signal that the brain understands. And so these different receptors that we have are going to be specialized to respond to a specific type of stimulus. Now, this should be real obvious to you because right now you know, for example, if I put a brownie in front of your face and put it right here, your eye would not be able to detect what the brownie tastes like, right? Your eye can look at the brownie, and the light reflecting off that brownie and being absorbed by that brownie might tell you that is a brownie. You can put it under your nose, and the aromas coming off that brownie might tell you that that is a brownie. And when you put that onto your taste receptors on your tongue, you'll know that that's a brownie. All right? And if it's directly out of the oven, you know, your thermoreceptors in your skin will tell you whether or not it's warm. I'm hungry. But notice that none of those receptors can do multiple things. They're each designed to recognize a single modality or a single type of stimulus. Now, there are many different types of receptors. This is not a complete list, but it is a list nonetheless that should help you move on to the next level. We have chemoreceptors, which, surprise, surprise, respond to specific chemicals. We have mechanoreceptors, which respond to mechanical change or mechanical energy. Thermoreceptors, sensitive to heat. I put cool up there, but just remember what is cool. It's just the absence of heat. So differences in temperature. Photoreceptors respond to visible wavelengths of light. Osmoreceptors, solute concentration. Nociceptors is the really weird one. This is a pain receptor, noci for noxious. All right. So we're talking about tissue damage, and there's different types of them. So some can recognize pinching, some can recognize burning, distortion, so on and so forth. All right. And as I mentioned on Tuesday, we have multiple types of perceptions that have no specific receptors. Instead, they're combinations of the different receptors. And so the example I showed that I described was the wetness receptor, and these are the different types of receptors that give you the perception of wet. It would be a touch receptor, a pressure receptor, and a thermal receptor. So different types of receptors are needed in order for us to understand our environment. And the way that a receptor works is no different than what you've already learned with regard to the neuron. All right? What we're going to do is we're going to create some sort of graded potential. All right? And so this process of creating a graded potential is a result of a transduction event. In other words, we're going to take a particular modality, a particular type of stimulus, and we're going to open up a channel to allow for an ion to pass through that channel, change the membrane potential, and if it reaches threshold, it's going to result in an action potential. Now, there's two different ways that we can do this. All right, this graded potential, I should point out, is called a receptor potential. Real fancy name. All right, and there's basically two ways. You can have a cell that is a receptor cell, all right? And so the stimulus is received by that, and then when that thing reaches threshold, it will release some sort of chemical messenger that will then bind to a channel in the afferent fiber that would ultimately result in the action potential, all right? Or what you'd have is you have your actual afferent fiber with the receptors located in the afferent end, and then so when you get that stimulus, that's going to cause a change in membrane potential that will ultimately result in threshold being reached, thus getting an action potential. All right. So either way, you can have an action potential created, just different mechanisms to do so. Now, typically what's going on is we're going to be seeing when we stimulate a receptor, it's basically going to result in an inward flux of sodium. So it's going to be a depolarization event. But sometimes we're going to see signal transduction in second messenger systems. So both ways work. And we're going to see, when we talk about the eye, we're going to see the less common. All right? So what we're looking at here basically is just a simple graded potential that ultimately results in the formation of an action potential. Now, when you look at one of these receptors, they actually are responsible for a very specific stimulus in a very specific area. We refer to this specific area as being called a receptive field. And so usually a receptive field is associated with a single neuron. So for example, that one neuron right there covers that whole area. And very often these sensory areas overlap. Now if we have a, let's see how to say this so I'm not confusing. Basically what you'll have very often is you'll have multiple areas that come through a single neuron that ultimately converge upon a secondary neuron. And this secondary neuron really is what is actually taking the input from all of these different receptive areas and gives you the perception of stimulation, regardless of which one of those particular areas you've actually stimulated. Now, if that didn't make a lot of sense, have you ever played that game with two pencils to say, can you tell me when I'm touching one area? And you do it kind of like this. Got to find things that are sharp. Always go to the Marine. All right, put your arm out. Okay, and put it up high so people can see it. so we're not going to actually do the game, but basically what you do is you put one caliper here and you sit with the other one and you say, okay, are they touching? And then you say, no, no, and then you slowly move in, and at some point you'd say,
ow.
No, you'd say, yes,
Yes.
yes, and then you'd look and say, oh my goodness, look how far apart they are, and everyone has a good jolly laugh. Right? Right.
Because the threshold is so close to the other.
Right, and so what that is basically is the secondary, or each of these receptive fields are basically overlapping and close together, so they actually stimulate the single secondary neuron. So each one is stimulating something downstream that basically combines those two signals. Now, did I leave marks on you? That's, well, I did that before I picked you. All right, now, if you did that on the arm and on the hand, you could basically find yourself bringing these things so close together that they're almost touching each other. In other words, the receptive fields are very, very close. But if I did the back of the leg, I could be about this far apart, and you would be going, oh, well, you know, yeah, they're touching now. And what this is showing you is that these types of sensory or receptive areas give rise to the degree of, or help us to get a sense of the world around us by having different degrees of sensitivity. Put another way, if I need to pick something up, I want to have pretty sensitive hands, right? I want to be able to feel and understand what I'm actually grasping, whereas it's not so important that I know where the spider is crawling up the back of my leg, as long as I know the spider is crawling up the back of my leg. Does that make sense? Okay, that's kind of what we're looking at here. We're looking at this, like this, is where you have receptive fields being very, very limited so that you have high sensitivity, whereas over here you have much, much larger receptive fields because we're basically using a secondary neuron to combine input from multiple spots. Yeah.
So, okay, if you're like drawing hot water through that, and you check the temperature of the water with your hand, you're like, when you get in, it's like,
Well, that's something else. Basically, it's just the presence of the thermoreceptors on your hand are a little bit, you have much more protective layers on the surface of your hand, whereas over here, it's a little bit not quite as callous. You don't have quite as thick a layer of corneal epithelium, or it's cornified epithelium. So, that's not so painful, right? So, in essence, what we have here is what I'm trying to get at is usually you're going to have your receptive field, which is associated with a primary neuron, and then that usually is going to synapse with the secondary neuron. But the degree of synapsing with that secondary neuron will give you the sense of how large a receptive field is. So in this case, we have lots of neurons synapsing on that secondary neuron, whereas down here, we don't. It's one-to-one almost. And so with this, we're recognizing stimulation at two different points, whereas this feels like one. Does that make sense? Yeah. Would be a lot closer together, right? Whereas, like on the back of your leg, not so important. It's not to say that on the back of your leg it's not sensitive, right? Because all you're trying to do in terms of touch and pressure is the getting to threshold. Once you activate threshold, that's enough to send a signal forward. But when we're talking about the fine control or the fine degree of isolating where that stimulation is actually taking place, back of the leg is not so important. Right? Whereas, say on your fingers and on your hands or on the bottom of your feet, it would be kind of important to know. Right? Because when you step on a piece of glass, it's kind of important to know where that glass is actually penetrated through the skin. All right? Now, one of the things that we need to understand, and this is not difficult to get, but it may not be something that you think about easily, is that when we're converting stimuli from whatever energy form it is into an electrical signal, we have to conserve whatever that stimulus is within the message of that signal. All right? Problem is we're dealing with an electrical signal, right? It's an action potential. It's, rest or it's a peak. That's it. There's no modality or magnitude other than that state. It's either resting or it's at a peak. And so in order for us to understand the difference between, say, I don't know, holding a piece of ice in my hand versus hitting my hammer or hitting my hand with a hammer, is going to have to be preserved within the context of the action potential, right? Because if I hit you with a hammer, that signal is being sent to your brain. Is it just action potentials? Or does your brain know that it's a hammer? Does your brain understand the concept of hammer? No, it only understands action potential, right? And if I take and give you a handful of ice, and you're sitting there holding, well, let's make it dry ice, because that's freaky cold. And you're holding dry ice in your hand. Does your brain understand dry ice? No, it just knows action potentials, right? So the modality has to be somehow preserved. And the way that it's preserved is through which receptors are being activated. So a pain receptor will only receive pain information. Temperature receptors will only receive temperature. And then what happens is those receptors are stuck, which in terms of the neuron, is stuck within a particular pathway. They never cross, right? You don't mix signals. So pain is always within the pain pathway. Thermoreception is always going to be in the thermoreception pathway. And this neuron that's traveling into the central nervous system is only carrying that information, and it's only going to go to a specific location because it's destined to go to that one place. There's no crisscrossing and mixing of signals. You do not taste purple. All right? So the stimulation of a specific receptor will always result in the perception of that stimulation. All right? Now, a little aside. We can be mad scientists. You want to be a mad scientist? Go get your PhD and then find out how much you get paid. No, I mean... I'm having fun today. No. All right, we're going to be a mad scientist, and what we're going to do is we want to be able to taste light. Doesn't that sound cool? So all we got to do is we just got to go into the nervous system, and we just got to find the optic nerve, and as it enters into the brain, we just have to cut it, and then we have to find the nerves that lead to the gustatory cortex, and then all we got to do is fuse those nerves together. You see what we've done here? We've created a chimera. All right. Now, is this easy to do? No. But they actually are doing, I think there's a sidebar in your book that actually talks about this is one way that the blind can actually see, is they actually taste light. They use a machine that takes light, and they're using the taste receptors to stimulate the optic nerve. It's kind of cool. All right. And what you're doing basically is because you've crisscrossed, because you become the mad scientist, right? And you've created this nerve that normally would go from the optic center to the occipital lobe, and what you've done is you've cut it, and now you've connected it to the fibers that are found here in the frontal lobe, is you're actually stimulating what you normally would recognize as being taste. So when you see red, I don't know, you're tasting waffles? Is that what I heard? You see my point. Does that make sense? So let me go here and then I'll come over here. All right, so I saw him first. Yeah. That's a good question. Well, all we have to do is do a real simple energy calculation, right? Of course, I don't remember anyone. Yeah, see, you're asking a physics question. Yes, ma'am. No, if in that case, what would happen if we were playing our mad scientist role? Remember, this does not happen really. We have to be mad scientists to do this. So put on your white lab coat and yeah. No, if you did that, you would no longer be able to actually use the gustatory centers to actually taste. The taste buds would no longer have a destination, right? No, well, except now every time you see some, we could cross them. In other words, we could take the optic nerves and send them to the gustatory center, but we could also take the taste buds and cross them to the optic center. So every time you put, Pop Rocks. Pop Rocks are yummy. right? You put them on your tongue, and the next thing you know, you're seeing all sorts of fun things. Grape equals, whatever. I don't know. Yeah, he did not die. That's an urban myth. All right. So modality is, you understand, so modality is preserved within the pathway, is what I'm trying to get at. So if you stimulate a optic receptor, in other words, a light receptor, photoreceptor, that information is destined to go down the optic pathway to get to the occipital center to let the brain know that light has stimulated that photoreceptor. All right? If you stimulate a touch receptor here in this finger, there's a pathway that travels all the way up and goes to the somatosensory center and says that receptor has been stimulated. All right? And that's the way we preserve the modality. There's no crossing over or mixing of signals. Yes. I'm not aware of any, but that doesn't mean that there aren't any, because as far as pathology is concerned, I know like 6. I mean, within all the grand scheme of pathologies. One of them is tonsillitis. I know that pathology. So, terrible. All right. Second thing is location. And actually the way that things are organized within the context of the CNS, the central nervous system is not just a mishmash of neurons. We even describe it. We said that the cortex itself is actually organized in some very discrete columns. And in fact, if you go into the central nervous system, we're going to see this a little bit later, is that the organization of the central nervous system is so well organized that we can actually stimulate it by, simply poking it and actually have the brain perceive stimulation in particular areas. All right? So the organization of the central nervous system gives rise or helps us understand where location is. So we send a signal down a particular pathway, and it goes to a very particular location within the central nervous system. And lastly, it's intensity and duration that gives us a sense of magnitude. All right? Remember we said graded potentials contain within them their own magnitude. So if I have a strong signal, that's going to result in a large graded potential. And if I have a long stimulus, it's going to result in an elongated graded potential. But an action potential has no magnitude. It's just on or it's off. And so the way that we code the intensity of the graded potential is through a series of increasing the rate at which action potentials are firing. And the duration is going to be with regard to how many or how long we're going to fire those action potentials. So magnitude and duration are encoded. You can think about it like this. It's like Morse code. No, it's not one, dot, dot, dash, dash, but Morse code is basically an encoding of a letter. Anyone who knows binary, and I don't know binary, it's an encoding of all sorts of signals. And all you got to do is look at the binary code and you go, okay, well, that's what this means. And that's what we're doing, is that the brain receives the action potentials in terms of how many, how frequently, where is it going, and under which track is it coming. And so then that's how it knows what's going on. That make sense? Yeah.
When you're talking about location, are you talking about not just the organization of the CNS in terms of where that stimulus is being mapped to, but the location of where the stimulus is relative to?
Well, so where it's mapped to in the CNS is going to be dependent upon the region that area from. It's going to be dependent upon which region it's responsible for receiving information from. And so what we're going to see in a couple of slides, we're going to see what are called the somatosensory homunculus and the motor homunculus. And really what that is, it just shows you which parts of the brain are responsible for which part of receiving from where, or which parts of the motor cortex are responsible for regulating which part of the muscles or from which parts of the body. And actually, you have an auditory cortex that's organized in much the same way. The gustatory cortex is organized in the same way, so that stimulation can be mapped very easily to a very specific locale. So we can say, for example, your big toe, anything that you receive from your big toe will always map to the exact same spot within the cortex. Sensory information coming in. Exactly. So you can think about it this way, and we'll come to your question here. Being the mad scientist again, we can go in there, take those neurons, cut them, cross them, and now when I smash your big toe, You don't perceive it's the big toe. You now perceive it's the ear. There you go. Kind of cool. How many of you guys have heard of phantom pain? Lose a limb. All right. Why? If I remove my limb and I can feel my fingers itching down here, why do I feel my fingers itching down here? The map is still in my brain. And so if I have threshold that's, let's say here at this stump, that's not an appropriate word, but we're just at the stump, I can't remember what the point of amputation, where I have a fiber that would normally go down and terminate, it's terminating here. And so while I'm stimulating here, the brain still perceives that that thing is mapped down to here. So that's why you feel the phantom itch or the phantom pain. Not necessarily, because usually it's because the way that the neuron has actually redeveloped or established itself, it's actually misfiring. So it's not, what you're doing is you're perceiving something that doesn't exist. And so it's not like an itch that you can scratch, it's just a stimulation that your brain's going, this is here, which is why it's frustrating. You had a question, and then we'll go over here. Mm-hmm. Yeah, it'll become diminished. And so actually the other thing is so, and again, it's with understanding the homunculus, let's say my fingers represent a small portion of the brain. Let's say I cut a finger off, and so now I have these two. Well, the area that was responsible for receiving this information becomes diminished because it doesn't really receive all that much information, whereas the area that's responsible for these will expand over, but it doesn't mean that this is lost forever. That's why you still get those phantom. Mm-hmm. Yeah, that's what I'm saying. So what will happen is that it will slowly take over, and this will actually start expanding outward, and this one will expand outward, and that's why you end up with more sensitivity in these regions. Does that make sense? Well, probably it's going to, well, because the opposite arms are going to be on opposite sides. That's what I'm trying to get at. So it has to do with what's actually localized around that region. The brain doesn't come along and go, that area I'm not using, so I need to do something with this. It has to do with the real estate that's around it, because everything is organized in strict columns. You had a question? Not anymore? No?
I know how different technology works, but like when it tricks your brain,
Mm-hmm.
It's like it's saying
Right.
I mean, how exactly is that going to use it?
Well, simply put, it's just about perception, right? The idea is that your brain is trying to make sense of the world around it given the limited amount of information, right? Your brain doesn't like not understanding what's going on. So, I mean, just think of anything. If you take a look at a picture that has been taken, it's a shot of somebody upside down, but it looks almost right, and your brain sits there and tries to fumble with it for a little while. I mean, this is the definition of an optical illusion, right? Your brain sits there and says, there's something inherently wrong with this picture. I know it's wrong, but the world around me is supposed to make sense, so I will make sense of this. I mean, the classic ones I love, if you ever go online and do it, are the street artist who does three-dimensional street art and then takes the picture. So always in the picture it looks great because the camera angle is always appropriate, so it really gives it the 3 dimensions. But if you go out there and take a look at it, your brain's going to go, you're on drugs. Because this doesn't look right. And it's going to try to make sense of it. That's all it's doing. It's taking the information it knows and trying to convert it into something that it can... Right. Right. Now, one of the things that your brain does is that it's, like I said, it's trying to help us understand the world around us. And so in order to do that, it wants to make the information as clear as possible for itself. And one of the things that it will do is a process called lateral inhibition. And this example here is trying to demonstrate that. And so you can imagine here's a pin being pushed into the skin. Now, just like any time you're pushing an object into a structure that is tied together, you're going to create an invagination that is larger than the actual point of entry. All right. In other words, the pin pushes down and your skin dips in. That's what I'm trying to say. Right. Now, in our little example here, when the pin pushes down, B is the site, the neuron is going to be underneath that site where it detects that. But remember we said, we have other neurons that have different receptive fields, and each of those different receptive fields are going to be stimulated as well, to a much lesser degree, right? So if we're pushing down here and it's going down and it's creating a huge signal, just outside of that it's creating a much weaker signal. But all that information would be sent onto the brain, and the brain would sit there and look at it and go, you know what? I'm receiving information from all these things, ergo the thing that's poking me must be as big as that receptive field that's being stimulated. But that's not the case. What's happening here is we're having a very limited or weak, or I should say very small signal or small object that's pressing into the skin. So to accentuate that, we have a mechanism called lateral inhibition. And what lateral inhibition simply does is in the pathways that are further upstream, beyond in the secondary and the tertiary neurons, what we're going to see is we're going to see lateral axons that are going to synapse with the lateral neurons as they're traveling along the way. And they release inhibitory neurotransmitter. And the purpose of them is to suppress the signal that's coming from the weaker neurons. And so what ends up happening is that the brain only gets information from the strong signal, and because you're getting repression, you end up with this appearance that instead of it being this kind of nebulous, blobby signal, you get this really, really strong, powerful signal that comes from the single locus. And so your brain perceives a much, much more limited signal that's much, much more powerful. Does that make sense, sort of? Now, if you haven't seen, or if you can't see it up here, what we have, when we're going from here to blocking there, that's presynaptic inhibition. Remember on the test, and that's presynaptic inhibition. We're blocking what's occurring here. And all we're doing is we're increasing contrast. We're giving a sense that this is much more powerful than it is because we are blocking this, and so in contrast to what this is nothing. So it gives a very large signal. Look at that. There's a relative. All right. Primarily because it sneaks in between the receptive fields. That's how I think about it. Also, probably because if you're using a larger object, you're probably coming into contact with multiple fields which are sending multiple signals simultaneously. Yeah. There's communication between each of them, and so in a cartoon like this, it's trying to keep it really, really simplified. Right? It's trying to say, oh look, there's only one, but you can imagine there's communication going this direction, there's communication going this direction, so they're each feeding back to each other and giving a sense of what sort of signals they're sending. So remember what we said is when we talk about the nervous system, it's never simple, it's grossly complex, and so all the cartoons and all the things we're going to see are going to be as simplified versions as we possibly imagine. Yes, sir. Mm-hmm. Yes, we're going to look at it when we talk about the eye. Yeah, but I mean, this is true for just about anything, right? I mean, think about this. Have you ever had a cherry limeade? And cherry limeades rock, don't they? I mean, they have basically two flavors. They have cherry and they have lime. But of course, who works at Sonic? Yeah, who works at Sonic? 16-year-olds. The answer is always fast food. 16-year-olds. 16-year-olds are notorious for making horrible mistakes. Rats in your food. Wiping their hands with their nose before they put it, right? And so every now and then you'll get a cherry limeade where they actually accidentally put in the ocean water, right? But did you ever taste the ocean water? No, of course, you didn't notice it. There's a reason you didn't notice it. Because basically your brain is repressing that information. Yeah. Yeah. No, I mean, I'm being kind of facetious here. I mean, because ultimately within everything, and I'm going to use this example again, ultimately in everything, it's a complex mixture of multiple stimuli, right? And I use cherry limeade because it's simple. We can think cherry and lime, but we know that the flavor cherry and the smell of cherry is not just a single molecule. It's multiple stuff that give rise to that flavor. But using cherry as a single flavor, or and lime as a single flavor, if you put something small in there and the brain, it's unimportant that you know that it's there, your brain is going to just say no. And so that's actually processed again at the level of the CNS.
Is that why when you're drinking like mixed cocktails, you start tasting the alcohol less and less?
No, that's just a function of that's great bartending, and also probably if the bartender's smart enough, he's putting less and less in there and charging you more.
Even when you're mixing your own, and you can even start adding more vodka, and you're like, this tastes great.
I think that's more of a repression. I mean, not repression, but the system actually just, yeah. That's right. Yeah. I say NOSI, but I mean, it's a specific type of receptor. It's a pain receptor. All right, but there are different types of pain. We can have nociceptors that are polymodal. They respond to different types of stimuli. We have nociceptors that can respond specifically to mechanical damage, so they'd be mechano in with regard to their modality, but they're still sending off a pain signal. And then there are thermo pain receptors, so on and so forth. Chemical as well.
So when you feel pressure, the brain is supporting
Exactly. Yeah. Now we have two things that I want to kind of cross now. We have adaptation. This is not the same thing as sensitization and habituation, which is very easy to confuse those two things. Remember what we said with habituation, sensitization was we used the model of the slug and we slapped its eye stalk, right? We're really cruel. Well, the adaptation actually refers to how a receptor responds to a particular stimulus, all right? And so it's basically, when we say adaptation, we're saying we're going to see a diminished response, all right? So we have two different types of receptors. I mean, you're going to hear me say this all the time. We have two different types of receptors, This is regard to adaptation, all right? We have receptors that are considered to be tonic, all right? This will be the tonic receptor up here. Tonic receptors do not adapt, or if they do adapt, they do so very poorly. In essence, what a tonic receptor is doing is it's constantly responding to the stimulus in the environment. So, your muscles, proprioception, all right, where your body is in space and how tight or how strong those muscles are in order to maintain posture is important, right? You constantly want to know what is the state of my posture if I'm trying to stand up straight. Am I going too far this way? Am I contracting too far that way? So on and so forth. And so you're always testing and trying to determine the degree of contraction of the muscles. For example, with the muscle spindle fiber, or the degree of stretch within the Golgi tendon apparatus, which is another type of tonic receptor that deals with proprioception. All right. So in essence, what you're doing is you're constantly looking, and as change is occurring, that's going to result in multiple action potentials or less action potentials, depending upon the degree of change. All right. So more change you have, more action potential you have. Less change you have based upon the set point, the less action potentials you're going to get. But notice you always have action potentials. It's just constantly sending information on. It's like this. If you had a clock, it is a terrible example, but if you had a clock and it was telling you, it's a second later, 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 it's a second later. All right? That's tonic. It's constantly telling you what's going on. Phasic, on the other hand, adapts quickly. All right? And so what you do is you get basically an on signal, and then when there's another change occurs, you're going to get another signal. It's like an on and an off. All right? So it's only when there are changes in the modality. So if you look at the graph, it's not always clear, but basically what this is saying, there's no stimulation going on, then all of a sudden there's stimulation and it's being maintained. All right? So that's what you look at. No stimulation, now there is stimulation, and that stimulation is being maintained. And so you can see down here the action potentials that occur. When we turned it on, we got a series of action potentials, and as it's being maintained, the action potentials decrease in terms of time until it turns off, and in which case there are no action potentials, at least in this particular example. Here, when we get turned on, we get our receptor potential, and that results in a series of action potentials. So basically what we said is we've had a change in the stimulation. And then nothing happened for a long period of time until there was another change, in which case you got a series of action potentials. So it's giving you the changes that have occurred. It's, again, like having a clock that says, oh, it's 5:03. Oh, you want, it's 5:03 in 10 seconds. All right, do you see? It's when there are changes that occur that need to be reported, that's when you're going to have phase it. Now, the easy example to remember here is think about your touch receptors in your skin. How many of you right now remember you're wearing clothes? Now you do, right? But up until this moment, you put on your clothes in the morning, you're like, okay, I put on my clothes, and you've gone on throughout the day and you have not thought one lick about whether or not that those pieces of clothing is actually touching your skin. It wasn't until I reminded you right this very moment that you went through the mental checklist. Yes, 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 yes. I got it all. All right? You didn't feel, or you haven't felt your shirt on your skin all day long. Right? It's only when something seems to be gone awry that you start recognizing the changes that have taken place. I mean, awry can be multiple things. I mean, snag your shirt on a branch or something like that, then you feel it's like there's a change. But as you're walking around throughout the day, it's like, okay, that's what I expect. All right? Changes in modality. That's the phasic, yeah. That I don't remember. That's a good question. I don't know the answer to that. Tonic. Because right, but that could be a habituation. That could be habituation. Right. I mean, so all right. So we'll try to use our brains here to try to figure it out. All right. So when you're talking about a thermoreceptor is turned on within a specific range. Okay. And so, for example, let's say you put your hand on a hot plate. All right. And what happens is the temperature rises very, very quickly in your hands, and then that's going to activate a particular thermoreceptor. And so that's basically going to be turned on, and it's going to be maintained until that stimulus is absolutely removed. It's not going to just say hot and turn itself off and say, okay, you're fine. Yeah, it's still hot, still hot, still hot, and then turns, oh, now it's not hot. So it's probably, when you're talking about a thermoreceptor, that's actually really more of a pain receptor that I'm talking about, but I think once it's been activated like that, it's going to remain activated. So I would go with tonic. I could be wrong. I am not offended if you come to me and say, no, you're full of crap. I looked it up on Google. I saw 3 forums where they talked about it. Yeah. Or Wikipedia. Wikipedia's smarter than me sometimes. This shows you an example of that homunculus. Our somatosensory homunculus. You can see up there in the top where it kind of shows you the organization. We're not going to spend a lot of time talking about the homunculus, but what I wanted to show you on this is the pathways, the somatosensory pathways, and it's really simple. I mean, I'm not going to go into too much detail here. Basically, each of the neurons within this pathway are called like the first order, the second order, the third order, fourth order neurons, and you start first order at the site of reception, where they come into contact with the second neuron. That second neuron is the second order neuron. Some features that you should kind of be aware of is that with regard to the first order neurons, there's going to always be some sort of crossover event. And where that crossover event is going to be dependent upon the type of reception, somatosensory reception you're getting. So when you're talking about, you can look at the picture over here, fine touch and or proprioception, those enter into the spinal cord, then travel up to the medulla, and that's where they're going to synapse with the second order neurons, and the second order neurons are the ones that are responsible for crossing over and moving up to the thalamus. All right. Whereas when you're talking about, say, pain, temperature, or coarse touch, sorry about all the ink on the slide, those synapse within the spinal cord, they cross over there, and then they use the tracks to move up to the thalamus before they go onto the somatosensory cortex. So they're specific with regard to where and when they're crossing over and where they actually synapse with the second order neurons, depending upon type. All right.
So the fourth ones were, that was an example. That's pretty rare, right? So it's not synapsing in the blood, but that's what I do.
That's correct. You're going to want to actually synapse and be sorted there. Well, right. But I mean, so with regard to the course, that's real basic information. Like I'm being touched, whereas the fine touch is I'm being touched by a spider. Right? Or, I mean, we could do it the other way is, you know, I'm holding something soft, you know, and fuzzy and cute. That's not somatosensory, the cute part. But knowing that you're actually touching something, that would be coarse. And so that information goes to the thalamus so that it knows. Remember, what does thalamus tell us? It's not I think.
There, but we don't know what it is.
That's right.
Yeah.
And so that's why.
Right.
Right. Spidey sense. I like that. Your spidey sense is tingling. Yeah, we'll just use that for now. The spidey sense is the thalamus. All right. So that's really what we're trying to get at here is that there's, again, it shows you the degree of organization, and we're not touching on how organized this is. When you get on to, and I'm talking to those who are planning on medical school, I don't know about dental school, I presume it's true as well. In medical school, you have two classes that you will take, neuroanatomy and neurophysiology, that go hand in hand, and you walk through every single solitary track and where they go and what they do, and then you wonder why you ever went to medical school. Now, touch receptors, simply put, are mechanoreceptors. There's a whole bunch of different types. I'm not going to ask you to memorize them. All right, sorry. All right, I'm not going to ask you to memorize these. I'm not going to ask you which ones they do what. Just know touch receptors are mechanoreceptors. All right, they respond to various types of stimuli. And you look at these things, senses vibration, fluttering movements, or stroking movements, which is why different types of touches feel differently. All right, because you have different types of receptors to recognize those particular types of movements. Some of them are encapsulated. Some of them have free endings. All right, and so they give rise to different types of receptive fields. Some of them are deep, some are not. All right. Nociceptors, simply put, there are three different types. You've got mechanical, thermal, or polymodal. You see how I kind of repeat myself when you guys ask me good questions? I like that. All right. So mechanical is obviously mechanical. Thermal is heat. Polymodal is all different types. All right. Nociceptors do not adapt, which is probably why I was thinking of the hot, not adapting. All right. They don't adapt. All right. If you maintain constant, if I come and hit you with a baseball bat all day long, they don't just say, oh, okay, this is life now. All right. You've seen the people who've walked on hot coals, and they say, look at me, I can walk across hot coals, or the people who can chew glass, you know, and do stuff like that. Do you think their body knows that the body's getting damaged? Yes. They're just ignoring it. All right. That's done at the level not of responsiveness, right? I mean, everyone, you all knew this guy in high school, right? The guy who'd get the lighter and say, look at me. You know, oh, look at me, how tough. Do you think that thing wasn't burning him, and the nociceptors weren't going, you're burning your flesh? Yes, that was what was going on. All right. Now, the way this is done is through a whole variety of chemicals. These are just some examples. Substance P is the easy one to remember because substance P is substance pain. All right. Histamine, serotonin, prostaglandins, these are all different types of chemicals that can actually lower the thresholds for pain. All right. This is important, right? Why would we want to lower the threshold for pain? All right. Take my baseball. Pop him right in the head. All right? If I come up, start poking at that thing, am I doing your head any good, or am I doing it bad? All right? Yes, I'm doing bad. Basically, that lowering that threshold is to remind you, don't let yourself get poked in the head. after you got hit with the baseball. All right, no one gets hit.
So you're saying when they keep in coma, in that case, like if there's already an injury there, it's going to lower the threshold to get the load of effect and avoid further.
There you go. That's what I was going to try to get. I was going to say next, cut yourself open. Don't cut yourself open. Yeah. It's a way to protect the body because what is pain? That's what some people will tell you. Pain is your body telling you, you're killing me. Literally, that is what it is. Your body is saying, if you continue to do what you're doing, we will die. That's why when you run, that's what your body's saying. Stop. Yeah. Yeah, that's actually a tougher question. And I don't really necessarily know the answer why our bodies have chronic pain. I mean, in fact, I'm pretty sure that the people who actually treat chronic pain aren't entirely sure why chronic pain continues to exist. Maybe it's just a pathology. In other words, the system is firing because what you've done is you've lowered the threshold so low that it can't see stop. You know, but as to why it occurs, I don't know. You know, but it's a good question. Now, there are different types of fibers through which pain is actually transmitted. We have A beta, A delta, and C fibers, and you can see that the A beta fibers, these are large and myelinated. If you're a large fiber and you're myelinated, how fast do the signals go? Super, super duper fast. These carry mechanical stimuli. We have the A delta fibers. These are small and they are myelinated. They are responsible for specific mechanical and or cold stimuli, and they carry fast signals. And then we have the C fibers. C fibers are small and unmyelinated. They deal with slow pain. Dull, persisting pain. And again, you can go back to the baseball. If you get hit in the face with a baseball, does that hurt? Remember gym class when you got hit in the face with a volleyball or a basketball? And then for about an hour, what did it feel like? Throb, 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 throb. Alpha delta, the fast, sharp pain that you receive, ow, and then the long-lasting throbbing pain afterward is through the C fibers. All right? Now, there are two pathways that are going to be activated. It's a reflective pathway. This is at the level of the spinal cord. You know what we're talking about here. Step on a tack, lift your foot. All right? But then there are always going to be ascending pathways that give rise to our conscious sensation. And this is what we're talking about when we're talking about those yahoos who walk across burning coals. All right? There's a reflective response. If you go walk across burning coals, have you ever watched somebody walk across burning coals? Do they just like say, look at me, I'm standing on the coals? No, it's, I can walk. And then as they get towards the end, but they're not screaming at the top of their lungs because they're able to repress their response, their behavioral response. So, protective mechanism, bring about your conscious awareness of tissue damage. All right? What we're going to do, we're going to get a motivated behavioral response. All right? If something hurts, am I going to do it again? No. Do we usually have an emotional response to pain? Yes. Weeping and gnashing of teeth. That's right. And generally, this is going to be influenced by some sort of experience. If your brother, every time he saw you, punched you in the arm, and you know, the first time it happened and keeps doing it, it's painful, and then by the time you're 40 years old, and he comes up alongside you, how you doing, champ? And you're going to sit there and go, ah, even though he didn't really do anything. Because your experience tells you, every time my brother punches me in the arm, it hurts. Does that make sense? So our experience tells us how things, or how we should behave in response. Yeah, well, kind of. I'll give you an example. My son, actually, when he was 3 years old, went to the Children's Museum, and he saw like this slide or something. It was all metal, and this was in the middle of summer, and he put both his hands down on the slide. And of course, they were screaming and yelling and stuff. And now, whenever he has any hot food or anything like that, it's too hot. It could be like, it's not hot. Look, I'm holding it in my hand. It's hot! It's not hot. Huh? Someday I might. He would just stand up here and hide behind the desk, though. Last slide, and then we'll get out of here. All right. So analgesia basically is a suppression of the pain response, and there are two regions that are part of the descending pathway to block the pain response. This is the periaqueductal gray matter, so it's basically next to your aqueductal gray matter, and then also the reticular formation. And simply put, it's presynaptic inhibition again. What are we doing? Well, what we're doing is when we activate a pain response, we're basically releasing some sort of chemical that goes up and gives us perception of pain. Well, what we want to do is we want to bind to opiate receptors within the axon terminal of our afferent fiber and basically block it with our endogenous opiates. So what we're talking about here are endorphins and calpholins and dynorphins, which is kind of the reason why we do things that are painful for us, like running. Right? Because your body's telling you, stop, you're going to die. And it says, oh, man, I don't want to die. I don't want to feel like I'm dying. So then it starts releasing the opiates, and then you start liking that you're actually killing yourself. All right, so running's not your thing? Think about hot food. Right? What is hot food? Hot food is basically, chilies have a chemical, capsaicin. Capsaicin binds to thermal receptors in the tongue. Those things basically tell you the food is hot, pain ensues, running around, gnashing of teeth, drinking of milk and beer and whatever you can get your hands on. And then what do? More chili. Right? Because we release endorphins to relieve us of the pain. When we get back, we'll start talking about the special senses. You guys have a great weekend. Go enjoy us playing rice. I don't know if we're going to win, but go enjoy it.
Our body likes to get high.
It basically does.